Okay, hello everyone. This is Judy Hervey, and I am recording our anemia presentation, basically on how to read a CBC. So here we go. Let me start this. Okay, your PowerPoint that you're looking at has a little bit more color in it. I have done this so that it will actually record a little bit easier for us. But it should be the same one, same information. So here we go. Basically, when we talk about anemia, do you suppose we're talking about a disease or a causation of something else? Basically, it's the latter. It's that anemia is called by many different reasons, and we're going to look at a little bit of a review in the very beginning of what we're actually looking at with red cells. So here are our objectives: understanding and interpreting a CBC, define and classify anemias. As you can see here, many factors go into it. We've got demographics, we've got dietary factors, and poverty, sociological and physical factors. Where you live can also depend on if you're anemic or not, because many different entities cause anemia. So understanding blood, I think most of you can see here that the plasma makes up about 55% of total blood volume. Cellular components, 45% of total blood volume, which have white blood cells, platelets, and red blood cells. We know there's a liquid component, which is the plasma, and look at how much water plasma is made of. And then the cellular component that has all the cells that we're always talking about. And then there's about 5.5 liters in an average adult, give or take, depending on the size of the individual. Plasma proteins, we know what they do. Carry antibodies and nutrients to tissues, and it carries waste products away. So that's why if we have a problem with the plasma, we're going to have a problem with many other things also. 7% plasma are proteins. Our globulin albumin is 60%, albumin holding that plasma together, and clotting factors, and are all synthesized in the liver. And that's why you can see if we have liver problems, tons of problems with clotting factors, and tons of problems with albumin and where it is, how it's stored. And there it is. Albumin molecules are large. They help with colloid osmotic pressure, meaning they're going to keep the plasma equal with our other body fluids. And erythrocytes, again, this should be a review: oxygen to tissues, and it removes CO2. It gives blood its red color, so the paler they are, we probably have a problem with an anemia. And it can't undergo mitotic division. So basically, they have a lifespan of 80 to 120 days, which would be anywhere from 3 to 4 months. And here are our leukocytes. We know what those are. And our thrombocytes, essential for blood clotting. And there's some pictures there if you want to actually look and see what they are. A breakdown of leukocytes, which are our neutrophils, basophils, eosinophils, monocytes, and our lymphocytes. And this just kind of gives you a picture of how long they last. And you can see when our lymphocytes are under attack, we're really pretty sick individuals. They last for years, monocytes months, and it's got phagocytizing properties, which help us stay free of infection. But then the rest of them are really limited to days in the body. Okay, so let's talk about a little bit about hematopoiesis. Basically, it's the process where all blood cells are produced in the bone marrow. We have master cells that are stem cells that create all different types of blood cells. The granulocyte is a special type of cell that protects the body, and neutrophils are a special type of granulocyte. They take 10 days to produce and only survive for 3 to 6 hours. So we know that, macrophages and monocytes need to come behind it. We also know how to keep up with this. The bone marrow is constantly producing granulocytes and neutrophils. It's in continual motion to protect our bodies. And then we have what we call a G as in George dash CSF, a granulocyte colony stimulating factor. So I'm going to say that again. It's G as in George dash CSF, just like how State Fuller did, a granulocyte colony stimulating factor. And this is a protein that signals neutrophil development. So you can imagine if there's a protein, a deficit of this protein somewhere from some disease entity or genetic factor, we're going to have an issue with the red cells. So hematopoiesis, again, occurs in the bone marrow, produces about 100 billion new blood cells every day. Two kinds of marrow. I don't know if you remember this. This should be a review also. There's red and yellow marrow. Red is active and yellow is inactive. And most yellow in the body always consists of lipids or fat, and they're fatty. Active marrow is found primarily in the pelvis at 34%, the vertebra 28%, cranium and mandible 13%, ribs about 10%, and proximal portions of the humerus and femur 4 to 8%. You can see proliferation from an original stem cell on this diagram here to the multipotential stem cell to the myeloid cell that projects out, and our myeloids project out to our neutrophils, basophils, eosinophils, and macrophages. So what is going on when our cytokines are released? For the most part, and I think we've learned this several weeks ago, infection or an invasion of a microorganism or damage, which would be inflammation. And here is a picture that kind of lets you know each type of blood cell comes from common hematopoietic stem cells. And when the stem cells proliferate, they differentiate under the control of a variety of cytokines and growth factors. And remember, a long time ago, we talked about cytokines, and there are communicators. They direct a lot of traffic. Some hematopoietic stem cells will continue to differentiate into hematopoietic progenitor cells. And these progenitor cells interact with neighboring cells and are exposed to particular signaling molecules, again, our cytokines. Under the influence of our stimulating factors, again, CSF, the cells will develop into mature blood cells. And this is all assuming everything is normal. Development of erythrocytes, we cannot talk about anemia and not talk about erythrocytes, is going to be our erythropoietin that stimulates development of the pro-erythroblast. And remember, the blast always means the babies. So it stimulates that. We get that from the kidney. If we have a sick kidney, we're going to have trouble with this process. That is where erythropoietin is stimulated from. So then it becomes a normal blast. The nucleus shrinks and it's absorbed and hemoglobin's present. Or it becomes a reticulocyte, leaves the bone marrow and circulates in the bloodstream for 24 to 48 hours until it becomes an erythrocyte. A normal retic count is about 1% and is regenerated every 24 hours. So now again on the CBC, you're going to know what a reticulocyte count is and why is the doctor interested in a reticulocyte. He wants to know how the production of the red cells are happening. And there it is. Retic count is useful to indicate if new red blood cells are being produced. And it is, retic is short for reticulocyte, but they will ask, what's the retic count? Erythrocyte final shape is disc-like. It's not round, but more oval, actually. When hemoglobin synthesis ceases, it loses its ability to form hemoglobin at this stage. 90% of an erythrocyte's weight is hemoglobin. Hemoglobin allows transport. Hemoglobin allows blood to transport 100 times more oxygen than plasma alone. So even if we have a decent amount of plasma in the body, but our red cells are down, we're going to have a problem. Our role of a hematopoietin, again, healthy kidneys equal oxygen regulation. When oxygen levels in the body are low, normal healthy kidneys will produce the hormone erythropoietin and send that signal to the bone marrow to produce more. So here it is in a nutshell. Decreased pressure of oxygen levels in the blood decrease tissue oxygen tension in the kidney. Kidney will secrete this hormone, erythropoietin, stimulates stem cells in the bone marrow to produce more red cells, and red blood cells carry oxygen to the body. So here is a part of our hemoglobin chain. Hemoglobin is the oxygen-carrying protein of that red blood cell. A single red blood cell may contain as many as 300 hemoglobin molecules, increases the oxygen capacity times 100. Each hemoglobin is composed of 2 pairs of globins, and you can see them here, and 4 iron complexes. Each hemidisc can carry 1 oxygen molecule. So this is basically broken down as to what hemoglobin looks like under a microscope. Real-life counter epoetin, trade names are epigen, epo, erythropoietin, and procrit. Those are all trade names. Indications meaning we can actually give erythropoietin. Anemia and chronic renal failure, HIV patients who have anemia due to treatment with AZT, and in cancer patients who have anemia due to chemotherapy. Action, what does it do? This, again, trade names of epigen, epo, procrit. It stimulates erythropoiesis, which hopefully will stimulate a red blood cell formation. It can be given IV or sub-Q. We're not going to give it if the hemoglobin is greater than 10 because we don't want to slough that system, which absolutely could happen if we're giving a medication that's going to stimulate us to have this hormone. May not see an increase in red blood cells for 2 weeks, so it does take a while to happen. Okay, now we're going to talk about what we call red blood cell indices. And these MCV, MCH, MCHC, you're going to see on a CBC printout. And it all has to do with the size and percentage of hematocrit hemoglobin in the red blood cell. So our mean cell volume, it measures just the size of the red blood cells. Are they big or are they small or are they average size? A mean cell hemoglobin is the average amount of hemoglobin in the average red blood cell. And then the mean cell hemoglobin concentration, it measures the amount of hemoglobin in an average blood cell in relation to hematocrit. So it's the percentage of red blood cells in a given area. And at that point, I'm going to stop this segment and I'll carry on in just a minute. So stay tuned for the next part.